MAGIC is actually an acronym for the Mount Sinai Acute GVHD International Consortium. And we formed MAGIC because we needed to accelerate the advances that we were making in the laboratory to bring them to patients. Graft versus host disease, or GVHD, is the major complication of allogeneic bone marrow transplantation. And it is one of the major obstacles for making transplantation safer and more effective. The GI tract is the critical target organ for graft versus host disease and its complications. So we now intensively study the GI tract in the laboratory, and we have cells, and we even have cells that will glow when they are alive. And we study the inflammatory reactions of GVHD in the laboratory very intensively. We bring that to animal models, and then we bring the best results to clinical trials in our patients.
GVHD that involves the GI tract tends to be the most resistant to treatment and the most likely to be fatal. What we have found is that the blood test that we developed, actually the biomarkers, concentrations are directly tied to the degree of damage of the GI tract. Our long-term goal is to eliminate steroids from the treatment of GVHD. And we think that we can find, for patients with high-risk disease, we can find combinations of drugs, most hopefully that are non-immunosuppressive, that will be more effective than the current standard. And for low-risk patients, we really want to have a steroid-free option.
The use of contemporaneous synthetic controls, as in the MAGIC database, is far superior to using the historical controls we've used in the past, because the use of historical controls has contributed to why we have not made progress in acute GVHD trials. And then the use of historical controls also suffers from the fact that clinical trials have eligibility criteria, and we're all more likely to enroll more stable, low-risk patients onto a clinical trial than what is seen or observed in historical control. After we've completed a multi-center single-arm trial in graft versus host disease, the best way to estimate efficacy or to understand if our results are compelling are to compare it to a contemporaneous control as we have in MAGIC. We're able to match control patients by many criteria because of the database and the number of patients that we have, and thus without taking the time to do a placebo-controlled randomized study, which is very difficult for many reasons, we're able to best estimate if our trial results show promise.
We are extremely happy about the long-standing cooperation with our friends and colleagues from New York, and fully support all research efforts. Within our institution, we have two main research areas. First, graft engineering and cell therapy, where we, for example, use regulatory T cells for the prevention and treatment of graft versus host disease. The second topic focuses on biomarkers and microbiome analysis in transplantation.
The MAGIC database provides real-time access to clinical data, and the integration of data from multiple international sites thereby excludes center-specific effects. Using the same assays and test validations at different sites ensures consistency across sites and availability of real-time biomarker assessment at MAGIC sites in Germany.
My experience with MAGIC has been transformative. I've been working closely with experts of bone marrow transplantation, and I've been exposed to a wide spectrum of research projects like basic science with mouse models and biomarker discovery projects, translational science, bringing the basic science knowledge to the patient and building computational tools to risk stratify the patients, and also clinical research with analyzing data of clinical trials.
It's been an amazing experience to have been involved with MAGIC since its inception, and we've already accomplished a lot, but we think that there's a lot left to do. And we've been making steady progress on a year-to-year basis, and that's very rare in medicine. And now we've hit this steep part where every year we're being able to make new discoveries and develop new approaches to the problem. So it's a very exciting time to be involved in this collaboration.
The work that we do here is really the tip of the spear. But what we're hoping is that as we validate our studies in more and more patients, in the US, more than 100 transplant centers already use our algorithm and biomarkers, and we're hoping that this is now going to be able to be even further disseminated throughout all transplant centers all over the globe.